Hey guys, we're going to be looking at Unit 1, Topic 2 today. So we're going to be moving on to multicellular organisms. Now in Unit 1, Topic 1, the focus was really on processes happening in individual cells, all of the different transport mechanisms, parts of the cell, and metabolism really. So now we're moving on to multicellular organisms and looking at how organisms function as a whole. And the first PowerPoint that we're going to be looking at today is on plant systems. Now, in terms of plant systems, we're going to be looking at gas exchange as one topic, looking at how oxygen and CO2 exchange through the plant, as well as transport systems in the plant. So how does the plant take up water, nutrients, and move it around to different parts of the plant? Now, in terms of your textbook, Pearson Biology 11, we're looking at section 5.4. It's quite a long section, so we're going to go through the PowerPoint quite quickly, and if you do the worksheet associated with this on the OneNote, you'll be well prepped for what you need to know on the topic. Okay, now there's quite a few curriculum descriptors that you need to know from this. So the first is describing the role of stomata and guard cells in controlling movement of gases. Explain how the leaf facilitates gas exchange. Explain the relationship between photosynthesis and the main tissues of leaves. So we're going to actually relate this back to what we've just done on photosynthesis and have a look at where this is actually taking place. Describe and contrast the structure and function of xylem and phloem tissue. So we're going to look at sieve tubes, sieve plates, and companion cells. Explain how water and dissolved minerals move through the xylem via the roles of root pressure, transpiration stream, and cohesion of water molecules. Discuss the factors that influence rate of transpiration, so things such as light, temperature, wind, and humidity. And explain the transport of the products of photosynthesis and some mineral nutrients via translocation in the phloem. So there's quite a few things that you need to meet here. We don't actually revisit this though, except for a little bit in Unit 3 when we look at ecosystems. All right, so when we refer to vascular plants, these are plants that have some form of vascular system, somewhat like humans where we have a circulatory system to move things around our body. Now this is most plants, so ferns, cycads, conifers, which are trees that produce cones, and flowering plants, which is most plants. Now these are called vascular because they have specialized tissues to allow gas exchange and the movement of nutrients and waste throughout the plant. Now we're going to start by having a look at the anatomy of the leaf, which is really important in understanding gas exchange and transport. So if we just take a look at the diagram and we take a cross section of a leaf, we have a similar top and bottom layers of the leaf, sort of like the top of the leaf and the underside of the leaf. And both of these sections are referred to as the epidermis, but we have the upper and lower epidermis. And within the epidermis, we have the very outer layer, which is the cuticle, and the guard cells, which surround our stomata, which are really important, and we'll talk about those later. But they're found mainly on the upper side of the leaf, and in some leaf species, they're found on the underside as well. That's sort of an adaptation that's different across different plant species. Now this big middle layer here is called the mesophyll layer, and there's quite a few different things going on in here. In this palisade area, we have the bulk of the chloroplast, which is where we know photosynthesis is going to be occurring, so the metabolic processes of the plant. Now in this other section among the spongy layer of mesophyll are our xylem and phloem, which are our vascular parts of the plant. So within our leaves, this is really where the transporting of materials is going to go. And it's not a coincidence that the chloroplasts are located in such close proximity because the products of photosynthesis are going to need to be transported via the xylem and phloem. So we'll come back to that a little bit later. All right, I'm just going to hide me here for a second. Gas exchange in vascular plants occurs in three main ways, and that's via the leaf primarily, as well as the stem and root tips. So we don't just think of the leaf with this. There is some gas exchange that happens in the stem and the roots. Now some plants aren't considered vascular and they're more simple plants like mosses. And the reason that they don't have a vascular system is because they're so small and thin, so simple diffusion of gases can just take place across the surface of the plant. So remember thinking back to topic 1 when we looked at gas exchange diffusion across a membrane. Where we have a very, very thin membrane, oxygen and CO2 are two very small gases that can diffuse straight across into the plant. But more complex plants, which are the ones we're going to focus on, need that vascular system to do that at the leaf, stem, and roots. Now, having another look at our leaf structure here. So very similar. They've shown the guard cell and stomata at the bottom here in this example in the lower epidermis. Now, stomata are little pores in the epidermis, and they're surrounded by the guard cells. So the guard cells are actually these big green cells. The stomata is this pore that opens up and allows gases to exchange. Now, when we did our prac looking at where we made our microscope slides taking leaf samples, you would have seen these guard cells. They looked like big, obvious cells that formed around the plant cells or in between two separate plant cells. And these would have been really obvious when we looked at them under the microscope. Now, the purpose of the stomata is to regulate gas exchange. They have to open and close according to the needs of the plant. Now, one downfall of the stomata is when they open, this is great to allow gases to come into the plant to allow for photosynthesis. But when they open, that allows water vapor to leave the plant as well. So sometimes we need to have a nice balance where we're still allowing enough CO2 into the plant, but without allowing too much water to leave the plant. Now, some small amounts of gases can pass directly through the epidermis and the cuticle via simple diffusion, but the bulk of the gas exchange happens in this massive pore known as the stomata. Now, it's not actually massive, but it's like a channel protein. Compared to just simply diffusing across the cuticle, it's going to be allowing a lot more gas to pass through. Now, this is a diagram out of your Pearson textbook, figure 5.4.2, and this is showing gas exchange in the leaf via the stomata. Now, this is really important because this is talking about the relationship between water levels in the plant and the opening and closing of the stomata. Now, you can see that in this example here where the stomata is open, when we have plenty of water available, the plant's able to take up that water and store lots of water in the vacuole, which we learned about in Unit 1, Topic 1. And when it stores water in the vacuole, this makes the cell turgid and it allows water to enter the guard cells, and that causes them to swell up and become enlarged. And when this happens, it opens the pore, allowing gas to enter the stomata. Conversely, if the plant doesn't have sufficient water, the vacuoles aren't full, the cell becomes flaccid, and that means that water is going to leave the guard cells and they don't have enough oomph to open up and allow gas exchange. This is an important mechanism because if plants aren't getting enough water, it's not productive for them to open the stomata because this is going to cause them to lose more water vapor. So it's counterproductive. In terms of the growth of a healthy plant, this is why watering the plant is important, because without enough water, the stomata won't open and not enough CO2 is going to get in to allow photosynthesis to happen. This is what the guard cells look like when we did our microscope work in the lab. We didn't look at it this zoomed in, but you could see the stomata sitting there quite obviously on the microscope slides. Another interesting thing is you can see chloroplasts inside the guard cells, and this is because they're going to be using energy to do this. And this is just repeating what I went through before. So when water passes into the guard cells, the internal fluid pressure increases, causing them to expand and open the stoma. Now, this happens, this is a really important point. The stomata open when we have plenty of water, when there's light available, and when CO2 levels are low inside the plant, suggesting that they need to open and allow more CO2 to come in. So typically the stomata are open during the day because that's when we have light available, and we know that when light is available and CO2 are available, the plant is able to do the light dependent reaction. Now at night, the stomata usually close because there's no light available. So the plant isn't going to go to effort to open the stomata and let carbon dioxide in if there's no light available and they can't do the light dependent reaction anyway. And that allows them to prevent excessive water loss, but that means that the rate of photosynthesis is lower at night. Now, in terms of the stem, you might have seen these structures before called lenticels, and we see them very often in mangrove trees. And if you look at the bark of trees, you'll often see these little pores in the stem. And they're able to take in gases as well as what we see with the leaf. Now, this is typical of woody stems. So instead of having an epidermis like we would have in a leaf, we have these cork cells, which are waterproof layers of dead cell that stop water loss and gain, but also allow gas exchange without the interference of water. Now, this is a bit more clear in the next slide. Now, if you look at this diagram in the back here of a layer of woody stem, we kind of have this corky layer on the outside, which is often brown, and it's basically dead plant cells, but they're there like that for a reason. They provide that nice waterproof layer. But the lenticel, if you have a look at this here, this is your barky dead cork layer, which is this part here. But the lenticel is a raised pore area of living cells, which are able to exchange gases. So going back to that previous slide, those little round pores are these lenticels, which are nice living parts raised up from under the dead cork layer that are able to take in gas into the deeper layers of the plant tissue. Now, in terms of the roots exchange gases with the air spaces that are within the soil. So having a look at this diagram up here, if we look at the roots, now the roots even go more microscopic than what's shown here, and zooming in, each individual root cell has a fine sort of root tip. We call them hairs. And thinking back to what we know about surface area, they basically are projections off the root cell to maximize surface area for uptake of water and gases. Now, soil quality is really important with this because soil needs to have sufficient air pockets so that gases can diffuse into the root tip, but also water can move through via osmosis as well. So it's important that when plants are watered, that they're not overwatered, or else we remove some of these air pockets and we can kind of suffocate the roots and make them rot. Okay, so this is just that other figure again from Pearson, but having a look more closely at what's happening in the leaf, the stem, and the root tip. So of course with our leaf we've got our stomata, which is the main mechanism by gas exchange in the leaf. In our stem we've got our lenticels, the raised pores of living tissue among the dead cork cells. And in our root tip we've got our root hairs that extend, and air can diffuse straight into those root hairs. Now, in terms of our vascular plants, there are two main processes that we need to go through. The first is transpiration, which is the process of water movement through the plant and its evaporation. The second one is translocation, which is the movement of sugars and nutrients from the leaf through the plant. So we're kind of going in different directions here. Transpiration is pretty much looking at water movement through the roots, all the way through the plant, through the stems and out through the leaves, whereas translocation is kind of the other way around. Photosynthesis happens in the leaf, producing sugars, and then those materials made in the leaf have to go from the leaf through the rest of the plant. So kind of working in parallel here. Now the transport of these substances, whether water, sugars or nutrients, these are going to occur in an area called the vascular bundles, otherwise known as like a vein, which are our xylem and phloem, what we talked about before. So the vascular tissue there is grouped as a vascular bundle, which includes the xylem and phloem. And we find these in the leaf, the root, and the stem. We looked at it here with the leaf, so we know what it looks like in the leaf, but I'm going to show you now what that looks like in the root and stem as well, because the proportions are a little bit different and the location is a little bit different. Now this is what the vascular bundles look like across the leaf, stem, and root. But first, let's just recap our xylem and phloem. Now, in terms of our transpiration, remember that's the transport of water and nutrients from root to leaf, bottom to top. That happens in the xylem. And the way you can think about this is xylem, X comes later in the alphabet, so lower towards the bottom to the top. Phloem are the ones going the other way, from the leaf, transporting sugars from photosynthesis to the roots. So going more from top to bottom, and that's translocation. So having a look over to the right, the vascular tissue is just bundled together here in the green. Now in the leaf, the vascular tissue is pretty much in the center of the leaf as it runs down the leaf. So if you kind of look at one of these pictures, that's your typical indented bit that runs down the center of the leaf. That's the major vasculature of the leaf tissue. In the stem, you've got these little bundles of vasculature that go around the circumference of the stem. So thinking about the stem being the length of the plant, you've got this vasculature going all around the stem, all the way up the stem, transporting materials either up or down, depending on the process. Lastly, with the roots, the vasculature is condensed in the center of the root, and that allows materials to just pass straight through the center in one location of the root. There's a video link there that you can watch in your time. Now this is just another representation of the same thing, except it's separating it now into xylem and phloem. And you can see in the leaf and the stem, they're both bundled together, but in the root, they've actually got a separate xylem and phloem. Because they're doing different purposes, they're different in the root cell. Now this is a cross section through a stem showing a vascular bundle with the xylem and phloem. So xylem are going in one particular vessel, and your phloem are in a different spot, and this is a stem, remember. So this would be looking at one of these little vascular bundles. All right. Now in the xylem, as I said, this is where transpiration is happening. So xylem from the bottom to the top. So this is the transport of water from roots throughout the plant. As well as water, the water also contains mineral ions obtained through the soil. So that's part of this process as well. Now in this process are obviously the xylem vessels, and we also have these structures called tracheids. And we'll have a look at those in a couple of slides' time. As the plant grows, the xylem matures into a long water-filled tube with elongated cells with very strong cell walls. So basically it's an elongated plant cell that grows long. The cell wall is very thick, but it actually dies and causes a big cellulose thick vessel structure, and it doesn't have a nucleus and cytoplasm anymore. So it's literally just cell wall with a hollow space for transporting. And that's what's shown there in that figure on the right. And you can see the arrows pointing upwards because it's heading from the roots to the rest of the plant. So the xylem vessels are made up of smaller elements called vessel elements connected together to form a long tube. And with our tracheids, these are singular cells connected through pits in cell walls. They're dead, they've lost their cytoplasm and nucleus, and have an open structure for water to flow through. This isn't particularly important, this is just looking at the structure of the xylem. Now in terms of absorbing water and solutes at the root, we talked about the root having this huge surface area because of the huge number of little projections of root hairs that come off the root. Now, water is able to simply diffuse straight across into the root hairs via osmosis because they're so small. But once they enter the roots, there's two different pathways for the movement of these ions and the water. Now, the movement can be extracellular. So a lot of the time, water and some ions pass between cell walls of individual cells. And you can see this over to the right in the red here. So water actually moves through the gaps between cells. And that's just easier than going through into the cell. In some cases though, it can be cytoplasmic, which is where substances enter the root hair, like so, and move straight through the cytoplasms of each of the plant cells. Now, it doesn't matter which pathway that the water and the solutes have gone through, we have to get the water and ions into the xylem tissue in order to get around the plant. So in the image on the right, you've got the root hairs here, which is what has done all of the absorbing of the water, and they're going to come in to this epidermis layer and deeper down into the endodermis. But then we have this layer here, which is called the Casparian strip. And that's a waterproof layer that separates the roots and the xylem. Now, because it's waterproof, it kind of is like our hydrophobic, hydrophilic interactions. It's going to cause the water and solutes that have absorbed into the plant to be forced into the xylem, where they can then continue up the plant throughout the vasculature of the plant. So transpiration is considered passive, like when we talked about passive transport. It doesn't require any energy, but it is assisted by 3 mechanisms, and I recommend that you watch the video to see how this works. Now, the first mechanism is the root pressure. Now, plants contain a large number of ions, and that creates a concentration gradient, like what we talked about in topic 1. So this means that water readily enters the roots via osmosis because there's generally a higher number of ions inside the plant cells than outside. And so that means that water will enter the roots because it wants to flow to an area of high concentration, and we create a high root pressure. This allows the water and ions to travel large distances up the stem because of that initial pressure at the roots. The second one is cohesive bonds, and this is talking more about the chemistry and the bonds between water molecules. And when we have heat energy from the sun coming down upon the plant, it actually helps to break the cohesive bonds between the water molecules and allow evaporation through the stomata. The last process is the transpiration stream, and this is where water molecules that remain in the leaf after evaporation actually stick to other water molecules from nearby xylem vessels to replace lost water. The way that this works is that we're going to get a pressure pulling water up the xylem vessels from the roots, and we get a one-way flow of water. So if we kind of think about the stomata opening, water diffusing out of the stomata and leaving the plant by evaporation, when that happens, water molecules are going to suck up to where the water was lost, and it creates this one-way flow where water move up the xylem and replace the water that was lost. And then if we think about it from a whole plant, we're going to get a movement of water from bottom to top of plant. Okay, now this is showing you this a little bit better. So we've got water moving into the roots by osmosis down here at the bottom. As it moves up, we've got water moving through the stem and moving into the leaf to replace water lost by evaporation. So when the stomata open here, we lose water, that creates a flow of more water to move up the leaf. On the right here, you've got some active transport happening when we take up salts into the roots. That has to happen by active transport, but we're not really going to go into that. But the uptake of water is via osmosis, and that water pressure, due to the difference in ion concentrations, creates that upward flow up the plant. So now we're going to look at factors affecting the rate of transpiration. And this is really important because transpiration goes hand in hand with our rate of photosynthesis. This is really important next year when we look at ecosystems, because it helps us to understand how all of these things work together to have an ecosystem working correctly. So the first lot of factors are environmental. Now, if we think about temperature, think about a hot environment and you've got plants out in the heat. If we have our stomata opening and taking in CO2 for photosynthesis, in a hot temperature, we're going to be losing more water through the stomata and we're losing water via the evaporation. So our transpiration rate is going to increase as the temperature gets hotter. Now, with humidity, this is an interesting one because humidity is talking about water vapor in the air. And so in a more humid environment, the rate of transpiration actually decreases. Think about it as a concentration gradient. If you've got a plant that contains a certain amount of water, and then you've got a certain amount of water vapor in the air, the higher the water vapor is in the air, the smaller the difference is between the air and the plant. And so that way, when the stomata open, if it's really humid, we're not going to be losing as much water through the leaf. So that actually decreases our rate of transpiration when it's more humid. Now, windy environments is going to increase transpiration rate. Now, just an important point, when I say increased transpiration, that doesn't mean it's a good thing. It means that it's increasing how much water is moving in and out of the plant. So when we've got a lot of wind and more air currents coming around, when the stomata open, water vapor is going to be really quickly dried out of the plant. It's going to move it away from the leaf and it's going to increase the evaporation of the water vapor. So often in really windy environments, plants need to take up a lot more water because they're losing so much with the wind every time the stomata open. Light is an interesting one because we know that light is required for photosynthesis. The more light that's available, the more transpiration occurs because when light's available, the stomata open to take in carbon dioxide for photosynthesis during the light-dependent phase. So the more light, the more stomata open, and subsequently that causes water loss. This is a diagram from your textbook showing these processes. So you can see some of those key things that I mentioned here. So here's our airflow. Wind increases rate of transpiration. Water vapor is removed by airflow, increasing rate of evaporation. Over here, heat from the sun increases transpiration because evaporation and diffusion are faster at higher temperatures. Humidity decreases transpiration because evaporation of water from the leaf slows when the leaf is surrounded by more moist air. We've also got physical factors that affect transpiration rate, and this differs depending on the plant, and different species have different characteristics about them that make them suited to their environments. The first is the number of stomata. So some plants have more stomata than others, and some more readily open than others. So the more stomata and the more they open, the more transpiration is going to occur. Different plants have different surface areas of their leaf. So with a greater surface area of leaf, we're going to have more transpiration happening. And lastly, the shape of the leaf and adaptations of the leaf allow different rates of transpiration and also can help to minimize the loss of water during transpiration. So for example, some leaves are folded, some are very flat, some have a waxy cuticle over the top to prevent water loss, and some have little hairs on the leaf surface. So the different adaptations that allow the plant to transpire at different rates. This is an interesting topic to look at as well for your student experiments. When rate of transpiration increases, the rate of water uptake increases at the same time. So as the plant loses water, it's going to take up water from the roots at the same time. If there's no water available or the roots of the plant are damaged, the rate of transpiration is reduced at the same time. So if we're not able to replenish the water that's been lost at the leaf, then to counteract that, the plant is going to close the stomata. And when it does that, it reduces rate of photosynthesis because we're not taking in CO2. Thinking back to at nighttime, when we don't have light available, transpiration rate decreases because the stomata are closed, because there's no sun, so there's no point taking in CO2, and the air is also cooler, so that's going to prevent evaporation of water at the stomata. Now let's have a look at translocation in the phloem. So this is going the opposite way, from sugars in the leaf made from photosynthesis going through the plant. So phloem are going to transport organic solutes like sugars from the leaf to the rest of the plant. And there's a number of different components in the phloem, such as sieve tubes, companion cells, parenchyma cells, and sclerenchyma cells. Again, we're not going to go into the details of these because that's outside of the syllabus, but you can see those in the diagram over here, which has come from your textbook, of the structure of your phloem. Mature phloem are living, unlike the xylem. So these aren't made of dead cell walls. These are actually living plant cells that do this particular job. So the sieve tube is this elongated structure here, and they transport sugars by acting as a big channel. So they are quite similar to the xylem. They don't have any ribosomes and they don't have a nucleus. But in order to remain living, they have these companion cells next to them. These aren't that well known, but they're thought to regulate the activity of the sieve tubes. They help them to do their jobs. We also have parenchyma cells, which are these ones around the outer edge. They assist in transport at the finest branches at the ends of the sieve tubes. And then you've got the sclerenchyma cells, which aren't shown here, I don't think. Let me just double check. No, they're not, which assist in mechanical support. Again, not that important, but just to give you an idea. All right, now we've learned about photosynthesis and we've learned about how leaves produce sugars, and the material that the sugars flow through in the plant is called sap. So you might have heard about sap when you break a leaf off or you snap a stem or something like that off a tree. And sap is literally the sugars made from photosynthesis in a solution. So sap is pumped into the companion cells and flows into the sieve tubes via active transport. So this requires energy to do this. The high concentration in the sieve tube cells creates a gradient which forces water into the sieve cells by osmosis, which increases the fluid pressure. Sucrose can then be actively removed from the sieve cells and stored. So for example, at the roots, shoots, and developing fruits where sugar is most needed. Now we can see this process over here of the xylem and phloem working together. Now you can see that the xylem is going from the roots up the plant in the process of transpiration that we talked about earlier. But some water is able to diffuse by osmosis straight into the phloem to assist this process. Now here's our leaf, which has the sugars in it, and the sugar is going to be able to move into the companion cell and into the phloem. It's going to move down because we're going from leaf to other structures. And via active transport, it's able to go into other cells like roots, shoots, and really high growing parts of the plant. Okay, so that's just a summary of what's happening with our xylem and phloem here. Water and minerals going up the xylem, and you can see that they're traveling in this direction. And photosynthetic products going from the leaves to areas that need it. So that could be the roots if it's a plant that's growing under the ground, so like potato plants, things like that. It might be up to the developing parts of the plant like the fruits that need the sugars. And again, over here on the right, here's our xylem and phloem doing their jobs. Now, thinking to photosynthesis, I mentioned earlier that this palisade layer of the mesophyll is where all of our chloroplasts are highly condensed. So that's where our sugars are being made. Now, this is just going over the roles of the leaf structures in photosynthesis. So the upper epidermis secretes waterproof, waxy sort of substances to prevent water loss. There's no chloroplast in there, but it's a see-through layer so that light can go straight through the cuticle into the chloroplast beneath. The mesophyll here is where photosynthesis is occurring. So you've got those palisade mesophyll cells, which are really packed closely together and full of heaps of chloroplasts. Got a typo there, sorry about that. And you've got your spongy mesophyll cells, which are down here, and they're loosely packaged, and they're going to allow nice gas exchange to be happening with the xylem and phloem. We've also got our vascular bundles that we talked about here. And our lower epidermis down the bottom is typically where we've got our stomata. Sometimes you'll see the stomata in the upper layer as well, which are really important for the gas exchange part. If we think about that, we need CO2 to be coming in the stomata, being able to make their way through this spongy mesophyll layer and diffusing into our chloroplast to do photosynthesis. Then our sugars are able to follow that translocation process via the phloem. This is just a summary of the leaf structures and their important parts. You can have a look at that in your own time. So there's a number of things that you need to know in this lesson. So you need to know the role of the stomata and the guard cells. That's really quite an important part of this PowerPoint. You need to be able to talk about the features of the leaf that allow gas exchange, the relationship between photosynthesis and those tissues of the leaf that I just went over. Be able to talk about the function of the xylem and phloem. So just know the differences between them and what they're doing. Explain how water and minerals move through the xylem in those processes that we talked about with root pressure, transpiration, stream, and water cohesion. Discuss factors influencing rate of transpiration. That could be a really cool one for looking at for student experiments. And explain the transport of the products of photosynthesis via the phloem. Now, in the Pearson Biology book, that's page 199 for your questions. There's also some handouts. Now, if you have a look in your class notebook, I have scanned a different handout, which is a biozone handout, which I would like you to have a look at and complete in the class notebook.